We're moving into chapter 9, and the focus here is on skeletal muscle. And you can see that here in the background image. We're looking at a skeletal muscle fiber, but we'll introduce the various types of muscle tissue and then move into the specifics with skeletal muscle and further into the muscular system as a whole. So to begin with the primary types of muscle tissues that are found within the body, we have what's known as cardiac muscle, and it's unique to one organ only, the structure of the heart. So looking at the wall of the heart, this is the only location in the body where we find cardiac muscle. And of course, this is what drives the pump action, the force behind our blood pressure. So that's first type here, cardiac muscle, unique to one organ, exclusive to the heart. The second type of muscle tissue, smooth muscle tissue, it's in various locations. And some examples would be the erector pili muscle in the skin, pulling on the hair follicle, the iris, which of course regulates the size of the pupil on our eye. And beyond that, many hollow organs, such as arteries and veins, you notice here. And in the wall of numerous hollow organs, arteries, veins, the stomach, intestines, there's a layer of smooth muscle tissue in their wall. And as we mentioned now, the focus now is more on skeletal muscle. And this is the muscle tissue type that makes up our muscle organs and thus the muscular system. And a principal trait of skeletal muscle tissue is connects to tendons, and typically the tendons connect onto our skeletal system, our bones. So that's the usual case, and we'll see some other exceptions, but typically it's skeletal muscle, tendon, and in turn connecting to bone. And it's this type of muscle tissue here that makes up our muscle organs, such as biceps and triceps and quadriceps and hamstrings. And of course, going back to chapter 3, the histology section, that's where we first brought up this information. And we just want to go over this again. So all muscle tissue has this unique ability to contract, to shorten the length of the cells. And with that contraction, with that shortening, it's producing a force. So generating this power that allows for some type of movement. So we just introduced the three types: skeletal muscle, cardiac muscle, smooth muscle. The locations of these, of course, skeletal muscle, as we just pointed out, primarily attached on to our bones, so via that tendinous connection. Cardiac muscle exclusive to the 4 chambers of the heart. And smooth muscle, again, many hollow tubes. The digestive system's a great example. So multiple locations for smooth muscle. And of course, with this force, skeletal muscles now moving our skeleton and our flexible joints. Cardiac muscle, this is the force behind our blood pressure as the heart contracts. And smooth muscle allows for various applications. The process of urination as we contract the bladder. Of course, delivery of a baby with contractions in the uterus, regulating the diameter of blood vessels. So that force applied has many different applications, unique to its distinct organ. And a final trait about the 3 classes of muscle tissue, you notice here how it's controlled by the nervous system. So of course, the brain, the spinal cord, the control center for the body. And when it comes to skeletal muscle, this is the only type of muscle tissue that has what's known as voluntary, or you could say conscious control, whereas all three types of muscle tissue has this indirect, involuntary control by the nervous system. So to give an example, what's meant by this voluntary control of skeletal muscle, at any time if you just decide that you want to wave your hand, flex and extend your fingers, wiggle your big toe, you have that direct, active conscious control to contract skeletal muscle at will. However, now this involuntary type of control, which again applies to all three types of muscle tissue, this would be at the unconscious level. And there's definitely times where skeletal muscle will contract without your direct conscious control. A great example is breathing. Your diaphragm muscle, composed of skeletal muscle, and you're breathing when you're unconscious, when you're asleep, or even throughout the day without any conscious thought put into that activity. You have muscle cramps and spasms or other examples of that involuntary contraction. Cardiac muscle has now only this involuntary level of control. So you simply can't just sit in one place, being inactive, and decide that you want your heart to beat faster and stronger. However, your mood, your behavior, your level of activity, if you're working out, so you're working your skeletal muscles, and the indirect effect, of course, is now to increase your heart rate and strength of contraction. But we don't have that just direct voluntary control. And the same applies for smooth muscle. You can't just simply decide that you want to dilate or constrict your pupil. But again, with changes in light level, then you have that involuntary level of control, that indirect level of control. And we'll learn much more about this voluntary versus involuntary when we study the nervous system. So we see the three types of muscle tissues, locations, how they're controlled by the nervous system. And again, referencing chapter 3, figure here describing locations, functions of each type of muscle tissue. And we see here another trait about cardiac muscle. Again, this unique type of cell junction. So between cardiomyocytes, binding adjacent cardiac muscle cells together, this unique type of cell junction, only seen in cardiac muscle, known as an intercalated disc. And with the figure here, this is a nice introduction, but we'll see the histology of these different types of skeletal muscle, cardiac muscle, and smooth muscle tissues soon. So now we're going to focus on skeletal muscle tissue, because this is the type of muscle tissue that makes up the muscular system. So moving forward, the focus will now just be for skeletal muscle with the remainder of this lecture and chapter 9 in your textbook. So seeing functions of skeletal muscle tissue, this first one, just movement to the body. Again, this typically connects via tendons onto our skeleton. So we're able to move our flexible joints and move our body. With skeletal muscles along our spine, along our legs, we're able to maintain our upright posture. So standing up as a biped on 2 feet. So that's coordination of hundreds of muscles to allow for that function. Skeletal muscle does give some protective supporting benefits to our body, especially in areas that lack bone structures. So a great example is, think of the abdominal region between the rib cage and the pelvis, and our abdomen, anterior and lateral sides lacking any bone features. And our various abdominal muscles are supporting our internal abdominal organs. The inferior floor of the pelvis, again, there's a large pelvic outlet, and that opening supported by skeletal muscle. An orifice is truly an opening, either an entrance or an exit point into the body, in or out. An orifice, such as your mouth, is controlled by skeletal muscle that allow you to open or close your jaw. You could flare your nostrils, and that's a function of skeletal muscle. The process of urinating, defecating. So many of these canals are controlled by these skeletal muscles that open and close a certain valve. And this final point here, skeletal muscles use a large amount of energy in the form of ATP. And as we consume and use ATP, that gives off heat. And with the tremendous amount of ATP that's consumed in our muscles, that's a source of much heat for our body. So now moving along, taking a look at a muscle organ as a whole, one single bicep is composed of thousands and thousands of cells that now all have to be bundled and bound together to build that larger organ. And connective tissue, we want to think of collagen fibers, elastic fibers. They're surrounding individual cells, bundling cells together to build the entire muscle. We'll start here at the surface, and we want to pay attention to the prefixes. This will really help us throughout this course. So the epimysium, this is the most superficial layer of connective tissue that's wrapped around the entire surface of the muscle. So directly at the superficial surface. So this is binding all of the deeper bundles of cells together. So now if we were to take a cross section through a muscle, and we'll see this on an illustration shortly, within the epimysium, there's now these smaller units, smaller bundles of cells. They're known as a fascicle. And you just see the definition. A fascicle is truly just a bundle of some type of cell. In this case, we're seeing a bundle of muscle cells. And each fascicle itself has its own layer of connective tissue. So bordering an individual fascicle is now the perimysium. If we now were to isolate an individual fascicle, it has multiple cells within. And then looking at one single cell, one individual skeletal muscle cell, right at the surface of each skeletal muscle fiber, the deepest layer, so you notice there, the deepest layer of this connective tissue is endomysium, surrounding each individual skeletal muscle cell. So if we work backwards, you have one cell wrapped in endomysium. They're all bundled together into a fascicle. That one fascicle supported by the perimysium. And then multiple fascicles bundled together. And the entire muscle organ is supported by the epimysium. So there's a large amount of connective tissue within the muscle structure itself. And we see this is now the site where the nerves that stimulate the muscle cells have to pass through these outer layers of tissue, and so do the blood vessels that nourish them. So let's take a look at this. We have this figure right at the beginning of the chapter, and we'll start at the small scale. So a muscle fiber, and this could be interchanged with muscle fiber or muscle cell. The cell membrane of a muscle cell is this term here, and we'll get to the cytology in a moment. But now wrapped around the entire cell, we see first that deepest layer of connective tissue, the endomysium. So now this entire unit here is brought together with multiple other muscle fibers. Each wrapped in its own endomysium. And then now the entire bundle, known as a fascicle, is now held together via the next layer, the perimysium. And now we take this individual fascicle and we could bring it right over to here. And now you see multiple fascicles, all brought together and supported by the outermost layer, epimysium. And we've now developed an individual skeletal muscle organ. And connective tissue is doing its purpose, binding and attaching the other tissues. And just look at this further. So the superficial layer, the epimysium, at the absolute surface. We could pull out a fascicle here. And now that's supported in the perimysium. Within one fascicle, pulling out an individual skeletal muscle fiber wrapped in its internal deep layer of endomysium. So now there's a large amount of connective tissue within the muscle that we're now aware of. But now that connective tissue actually branches off the extreme ends of the muscles. And that same collagen material blends right into the tendons that are now on the borders of the muscle. So a typical tendon, the typical description is connecting from skeletal muscle to bone. And you can see an example here showing this Achilles tendon from a calf muscle. The Achilles tendon is the common name for it, a calcaneal tendon. And here it is connected to the calcaneus, muscle to bone. However, there's other exceptions. In terms of facial expressions, it's truly the muscles are pulling right on your skin to allow for facial expressions, smiling, blinking, whatnot. And also there's examples where one muscle is truly connected to another muscle to fix it in place, to hold it in place. There's this term now, aponeurosis, which is truly just a distinct type of tendon, whereas most tendons tend to be these thin cords, such as this Achilles tendon. And aponeurosis, described as just a very broad, flat tendon. And a great example is right here on the superior portion of the cranium. So that's represented here on this clay sculpture. It's called the Gallia aponeurotica, or epicranial aponeurosis. Just an example of a broad aponeurosis. We have these over the abdominal region, the anterior abdominal region, also the lower back has aponeurosis as well. And a great example right here on the top of the head. And it has a similar function. You notice here, this occipitalis muscle connecting right to that particular aponeurosis structure. So we want to take a moment now to look at this table here that's in the textbook. And we have a lot of the information here on the screen. And we're seeing that there's now a little bit of a system when it comes to naming the muscles. The bones, truthfully, the names and the descriptions were just based on what people saw as they were discovering and naming these features many, many, many years ago. But for the muscles, there's a little bit of a system in place. So we begin here. Some of the various traits that are used to name muscles. One is the direction, the position of the muscle. So we have what's known as a tibialis anterior, and both parts of the names are really telling you where this muscle be located. But we want to start here, the really descriptive term here. Anterior, so it's towards the ventral or anterior side of the body, that tibialis anterior. Another example, there's muscles called serratus anterior, serratus posterior, just to give some examples. So the position on the body is one characteristic of the name. You can see here what's known as external oblique. Well, anytime we see the word oblique, that means something's running at an angle. And that is the position of the external oblique muscles on our abs. The rectus abdominis, the term rectus means straight. And right down the middle of our abdominal region, what we commonly call the six pack, there's actually 8 segments to it, but running straight down the midline of our abdominal region is the rectus abdominis. So direction is one characteristic that can apply to a muscle's name. Second one would be the region, the body part. So we can go right back to this example, rectus abdominis, because it's on the abdominal region. That six-pack ab muscle, rectus abdominis. Or another good example, extensor carpi ulnaris. Well, in the forearm and antebrachium towards the medial side, hence that ulnaris near the ulna bone. And also the term carpi, carpus would be the wrist. That's where it acts on. Another great example, just going right back here, tibialis anterior. It's running right on the anterior side of the tibia bone. External oblique, the meaning of external is something that's found in a more superficial location. There's an external oblique as opposed to an internal oblique, which is deeper. And of course, rectus abdominis, right on the abdomen. So as a whole, some of the many traits that go into naming a muscle, what can be used to name a muscle would be the shape. There's muscles known as orbicularis oculi, orbicularis, because as in orbit, a circular shape. The size, here's a great example. You've heard of this muscle called gluteus maximus, because it's the largest of the gluteal muscles. There's a gluteus medius and a gluteus minimus. Movement, so what's the actual motion that that muscle controls? And here's a great example. Extensor carpi ulnaris. This is a muscle that will extend the carpus, and it's located towards the medial side of the forearm. So extension would be the motion it does here. There's muscles called pronator teres or supinator. There's a muscle called a flexor hallucis longus because it flexes the hallux. So as a whole, the size, shape, the movement, the location, the direction, these are all some of the different qualities that go into the name of a muscle. But of course, keep in mind there's always going to be many outliers. So as a last example, with this in mind, we see this name here, biceps brachii. And what we're going to see are 2 branches bi that's on the brachium. So the 2 branches just anterior to the humerus, biceps brachii. So now moving through this section, we're going to start with the cytology of skeletal muscle tissue. So looking at its microscopic structure. And a helpful figure that we'll go to is this figure 9.3. And we're now just describing structures of a skeletal muscle cell. And you'll note that many times we're going to be using these prefixes, both sarco and myo. And both of these pertain to muscle. So as we start out, we could begin here with what's called the sarcolemma. Every cell in the body has a plasma lemma or a plasma membrane, a cell membrane as well. And we see just this specific term used for the cell membrane of a skeletal muscle cell is the sarcolemma. There's the sarcoplasm, simply just meaning the interior space of a muscle cell. All of the area inside of the sarcolemma is the sarcoplasm. And when it comes to skeletal muscle in particular, they are these long cylindrical cells. So we can't interchange the word fiber or cell when it comes to muscle tissue. So as I mentioned, these long cylindrical cells, that's the shape of a mature skeletal muscle fiber. And it also is a multinucleated cell. Along the length of that long cylinder, there are multiple nuclei in a mature skeletal muscle fiber. And how this develops is due to the fact that we have these precursor cells called myoblasts. So myo meaning muscle, and blast, of course, the younger, newly developed cell. These precursor myoblasts each contain a single nucleus. And as multiple myoblasts then bond together, they start to develop into a larger, now multinucleated cell. And this process continues several times over to make the mature, elongated, multinucleated skeletal muscle fiber. And here's a much better figure. You notice this picture 9.3. Each myoblast has its own single nucleus. They're bonding together to make a multinucleated cell. The cell membrane, known as the sarcolemma, and all of the interior space within the sarcoplasm. So now we notice within the sarcoplasm, we start to see now these bundles known as myofibrils. One skeletal muscle fiber is filled with, you notice here, just dozens and dozens of myofibrils. So going into a smaller scale, the myofibrils themselves are made of bundles of multiple myofilaments. And they come in the two varieties. You notice there's a thick myofilament and a thin myofilament. The thin myofilament is composed of three components. There's three parts to it. The principal structure of the thin filament, however, is a protein known as actin. Whereas now thick filaments, these are now just large bundles of a protein called myosin. And together, these two myofilaments are arranged in a precise manner to make the larger myofibril structure. But now we can notice that the entire myofibril itself is wrapped in now these other features, these other organelles. And that's where we'll touch on the sarcoplasmic reticulum, the terminal cisterna. And let's continue forward. So wrapped directly around the myofibrils, there's now the multiple sarcoplasmic reticula, commonly abbreviated to SR. And this is truly equivalent in structure to the endoplasmic reticulum that are found in a typical cell. but again, using that prefix for muscle. The sarcoplasmic reticulum, wrapping around the structure of the myofibril. And their key function is they store, and more importantly, they release calcium ions. And this calcium release is a vital step in order for the muscle cells to contract, to shorten, and to produce that force for motion. Bordering on the edges of a single SR are now the terminal cisterna. And we'll go back, of course, to the textbook figure. But right here is a sketch now of a sarcoplasmic reticulum. And the expanded edges on each side are known as the terminal cisternae. There's these expanded edges of the SR. And running right along the terminal cisternae are a network of canals, a network of tubes, known as transverse tubules. So if we add to our sketch, we'll put a transverse tubule boarding right along each terminal cisternae, and then we'll draw the next sarcoplasmic reticulum. And that leads us to this final trait here for this slide, a triad with 3 parts, of course, 3 components. A triad is now the union of 2 adjacent terminal cisternae and the transverse tubule that's running between them. So we'll highlight that right here. So that union of 3 parts, 2 terminal cisternae, a single transverse tubule, that union with 3 parts is known as a triad. And we'll go to the textbook figure once more. So we can start with the myofibril, composed of thin and thick myofilaments, all bundled in the calcium-storing sarcoplasmic reticulum with this expanded edges known as the terminal cisternae. Running between them here, highlighted in yellow, are the whole network of transverse tubules. And you can see that unit there of the triad. So going back to the description of T-tubules, we can see that a T-tubule, it truly is a connection between the sarcolemma, the cell membrane of a muscle fiber. They start at the surface, and now those transverse tubules penetrate all the way deep in, and they run right along the terminal cisterna. And that's truly their purpose. They're an ion channel that's connecting those 2 areas. An ion channel between the sarcolemma and the terminal cisterna. And just a closer look at that here. And even closer in. So starting at the sarcolemma, which leads directly into a transverse tubule, and bordering right against that terminal cisterna. So now we're going to take a look just at the larger scale view of what's going on here for muscle contraction. In order for a skeletal muscle fiber to begin its contraction, it needs to receive a signal from the nervous system. And this comes via a neuron known as a motor neuron. This is a cell of the nervous system that's signaling from the brain or spinal cord, sending this signal down to the skeletal muscle, ultimately to stimulate contraction. So in this first step here, we're looking right in this area known as the neuromuscular junction. It's where the nerve cell and the muscle cell communicate to one another. The nerve cell is releasing a chemical, this neurotransmitter. And for muscle contraction, the specific neurotransmitter produced is known as acetylcholine. And we'll use this abbreviation here, just ACH, standing for acetylcholine. It's released out of the nerve cell, and it binds to a receptor that's found on the sarcolemma, on the surface of the skeletal muscle fiber. Ultimately, this opens up ion channels on the sarcolemma. So now ions are able to penetrate into the muscle cell, and this leads to an electrical current. And ultimately, this electrical current is what stimulates the contraction function of the skeletal muscle fiber. And the details here, of course, that's now getting into the physiology. So we won't go any further than this. Just the production of the neurotransmitter ACH, interacting with the sarcolemma, and triggering the process of contraction, the shortening of the skeletal muscle cell. However, this is not a realistic portrayal to see one motor neuron communicating to just one muscle fiber. Instead, we have what's known as a motor unit. And a motor unit is described now as the total number of skeletal muscle fibers that are controlled by an individual motor neuron. And there could be variation in the size of a motor unit. The smallest possible motor unit would only have 3 cells, meaning that one cell is the motor neuron, and now the 2 skeletal muscle fibers that it's controlling. So here's a very small size motor unit. A single motor neuron is going to signal and release ACH on these 2 different skeletal muscle fibers, and stimulating these 2 cells to contract simultaneously, to work as a group, as a unit. Whereas the largest possible size motor unit would be 1 motor neuron that's controlling up to 2,000 skeletal muscle fibers simultaneously. The benefit here being a very small motor unit, just only controlling a few number of cells, could lead to very, very fine, precise motor functions. And think about the functions of your fingers. Like if you're typing or playing a musical instrument, you can have that very precise, controlled, subtle motion. Whereas a very large motor unit, contracting up to a few thousand cells simultaneously, large motor units could generate a lot of power, a lot of force. So now moving on to the structure of the skeletal muscle fibers, we'll go a little deeper in. And we'll discuss what's known now as a sarcomere. So just once again, seeing that prefix, sarco. This is now the unit of physiology, the unit of contraction. So once a skeletal muscle cell is contracting, it's truly the changes that go on within the sarcomere that actually lead to that shortening of the entire cell. This is related now to the previous structures we were just looking at, the myofibrils, themselves made of the thick and thin myofilaments. So myofibrils are now segmented into multiple sarcomeres. In fact, it takes about a few thousand, several thousand sarcomeres connected end to end to make just a single myofibril. So at this point, we're describing something that is extremely tiny, these sarcomere structures. But you can see the concept. If each of these sarcomeres has the ability to contract, and their contraction only shortens just by a few tenths of a micrometer, but with each sarcomere contracting, and this happens over the length of multiple thousand sarcomeres, that'll make the entire myofibril shorter. So a simple analogy is if you think of a chain is made of a series of links. So these links are connected together in series, just meaning end to end. So multiple links together make this entire chain. And if we can make each link shorter, then you can see how the entire chain would be shorter. Or another simple idea is think of a train. The entire train is formed by the linkage of several cars. So if you had shorter cars, you'd have a shorter train. So that's what we'll move into next now is the microanatomy of an individual sarcomere. And it's labeled with all these different letters here. You notice Z, I, M, H, A. We'll go through the various parts. And again, keep in mind the sarcomere is just one tiny little segment of the much larger, much longer myofibril. So we'll begin here. This is a good figure to start with. We'll begin with what's called, it could be called an I band or a light band. Those could be interchanged. You notice you spell the word light with the letter I, to help you remember that. The I band area, first of all, the I band actually is shared between neighboring adjacent sarcomeres. So it actually overlaps neighboring sarcomeres. And the I band, this only contains thin filament. There's never thick filament in this region. Whereas now the dark band, the A band, this is the area that contains all of the thick filament located here. So you notice the labeling here, the A band, and the purple lines here are representing the thick filament. The border of the sarcomere, right at the edge of the sarcomere, and it appears here as kind of this zigzag line. That is known as either a Z line or Z disc. And again, all of these terms can be interchanged here. This is now the border of the sarcomere. This is where one sarcomere is connected to its neighbor, is at the Z disc. And another trait is the thin filament. The thin filaments are now connected to the Z lines. And that's represented here as the red line, the thin filament. And the thin filament is always connected on to the Z lines. So these first few parts here, I band, A band, and the Z disc. Just a closer look. We stated that the thin filament is made of three principal parts. The actin is the principal structure. The major structure here is represented by these pink circles. However, wrapped around the actin, there's a second structure known as the tropomyosin, as well as the troponin. And we'll take a look at each of those. The thick filaments, again, the full span of the thick filament. That's located here in the A band that we covered earlier. Overlapping adjacent sarcomeres. That's the area of the I band. That's where we only have thin filament present. The Z disc, as we just pointed out, that's the boundary of one sarcomere is from Z to Z. And the newer terms we're adding in now, the M line. The M line is the center of the sarcomere. And the M line is the connection point for all of the thick filament. So the myosin thick filament is all centrally connected to that M line. And then lastly, the H zone. The H zone is a smaller area within the A band. And what's unique about the H zone, this is the area of the sarcomere that only contains thick filament. So represented here by the purple lines branching off of the M line. The H zone only contains thick filament, myosin, whereas the I band only contains the thin filament, the actin filament. So it's with all of these components here, there's the border of the sarcomere, the Z disc, the area that contains just thin filament, the I bands, the center of the sarcomere, the connection point for the thick filaments, the M line. All of the thick filament is bound within the region of the A band. Within the A band, the area that only contains thick filament is the H zone. So it's these 5 areas here. Taking a closer look at each of the myofilaments, there's the thin myofilament. And we, again, described how there's 3 components, and here they are. The majority is composed of actin. Wrapped around the actin is the tropomyosin. And in turn, connected to that is troponin. We'll go back to the illustration to see this, but the actin itself is now just physically blocked by the position of the tropomyosin molecule. And in fact, it's covering the active sites that are on actin. These binding sites are just physically blocked. As long as these active sites are blocked, the sarcomere and thus the muscle cannot contract. And that's the job. Tropomyosin is preventing contraction. Whereas this third protein, troponin, it's holding the tropomyosin in place. So it's aiding in the position of the tropomyosin, which of course is blocking those active sites. So the way to remove each of these is with calcium. So calcium directly interacts with the troponin. So this no longer holds the tropomyosin in place. The tropomyosin can now be released to expose the active sites on actin. So calcium is the source of removing both of these 2 proteins here. So we got to think about where it's the source of this calcium. And that is the storage site of the sarcoplasmic reticulum, which is directly wrapped right around the entire myofibril. And again, with the release of calcium ions out of the SR, calcium directly interacts with the troponin to remove tropomyosin. And now we could expose the actin filament. And we'll see that here again. So a thin myofilament, principally made of the actin, in a resting, relaxed muscle. Calcium ions are stored in the sarcoplasmic reticulum. So now we have the tropomyosin just physically bound to the active sites, blocking that location. And tropomyosin, again, maintaining that position. As we just described from the SR, there's the release of calcium ions that directly interact with the troponin. Leads to the disassociation of troponin and tropomyosin to expose these active sites. And this is now where the thick myofilament comes into play. The thick myofilament, made of large bundles of myosin proteins. The myosin, as we just discussed, is connected centrally in the sarcomere to the M line. And We'll sketch that out here. The myosin itself, the side connected to the M line is known as the myosin tails. And the opposite edge is known as a myosin head, sticking out on either side. And once the actin has been exposed, the myosin head has the ability to attach on to actin. Again, that can only occur after the release of calcium. So at this point, we can now have an attachment, a cross bridge between the myosin and the actin. The thick myofilament and the thin myofilament are now connected to one another. And we can see that here in the illustration. So with all of these multiple myosin heads, now with the potential to attach on to actin, the next step is now, we notice here on this figure, the myosin head has this ability to move at the hinge. So once connected, the myosin, you can imagine on this side of the sarcomere, attaches on to the actin and can pull that actin towards the center of the sarcomere. And this happens on both sides. So an isolated muscle cell, even an isolated sarcomere in theory, would pull both sides of the thin filaments closer to the M line, closer to the center of the sarcomere. And if we were to do that, that would reduce the overall length of the sarcomere. That would lead to the contraction of the sarcomere. And that's truly the theory of contraction. So we're just going to touch on the overall outcome. We won't get into the detailed steps at this point. So when a skeletal muscle is contracting, think of your biceps on your brachium as you're contracting to flex your elbow. Well, occurring at the cellular level, occurring within the sarcomeres. The I-band segment, that is reduced in length. The Z lines are being brought closer together. The length of the sarcomere is being reduced. The sarcomere is shortening. However, the thin and thick myofilaments stay the same length. So these elements are not contracting. It's just truly their position relative to one another is changing. Much more of the thin myofilament is getting pulled into the A band. More of the thin myofilament is getting pulled closer to the M line. And a description here, and we'll see this visually. So again, with the storage of calcium in the sarcoplasmic reticulum, that calcium's released out and interacts, of course, with the troponin, releasing tropomyosin. Myosin heads attach on to actin. Actin is always connected on to the Z lines. So we now begin by pulling the actin closer to the center of the sarcomere. And again, that connected actin always bound to the Z lines. When we're pulling the actin closer to the center of the sarcomere, we're also pulling those connected Z lines as well. And reducing the length between one Z line to the next Z line, reducing the length of the sarcomere, is contracting the sarcomere. And again, you want to extrapolate this to the fact that multiple thousands of sarcomeres all linked together make one myofibril. And again, if each sarcomere is contracting, the entire myofibril will shorten. And a great illustration here. So a relaxed sarcomere, a resting muscle, and we see this distance between Z line to Z line, the length of a sarcomere. And as we just described, myosin heads pulling actin towards the center of the sarcomere, dragging those connected Z lines, and we see that reduced length of the sarcomere. However, the length of the myofilaments, thin and thick, remains the same. The I-band area is reduced in size. And you also notice here, the H zone is also reduced in size as well. More actin is moving into the A band, reducing the size of the H zone. And here's just a nice animation kind of putting these parts together. So we'll go through this. You notice the color coding here on the side. As our sarcomeres are contracting, again, truly what's happening is the myosin is attaching on to the actin, pulling it towards the M line. And we notice here in red, here's our myosin molecule. This would be the myosin tail. The M line is off to this side of the screen. We have the myosin head, which you notice has this ability to move. That's connecting on to actin, represented here. And you notice by pulling and releasing and pulling, we're now just pulling that actin closer to the M line. Wrapped around the actin are now the troponin and tropomyosin. And to release them, this is the need for calcium. So we can imagine that our sarcoplasmic reticulum would be somewhere over here. And we're releasing this yellow little circle representing calcium. Calcium first directly interacts with the troponin. That now adjusts the position of tropomyosin. And it's only at that point the myosin head attaches onto actin. The final player that we are concerned with is now the energy source here. You notice in green ATP. So produced in large numbers in the cell, the mitochondria specifically. And every time the myosin head moves one direction and back, that's using the energy source of ATP for that activity. And you have to now extrapolate that over just millions of myosin heads using so much ATP, giving off so much energy, and adding to the production of heat from our muscles. This next topic is now looking at the shape of muscle tissue structures. And we see these various shapes all the way at the surface of the muscle organ. So depending on how the fascicles, the bundles of muscle cells, their orientation and connection to the tendon, we can see these 6 principal shapes, these 6 principal appearances for muscle organs. So we have the description here, and then we'll skip to a figure that shows these. So to begin, what's known as parallel muscle, the fascicles of muscle cells, they are arranged parallel to the length of the entire muscle organ. So a simple sketch, you have a tendon on one side, another tendon, and the skeletal muscle cells. I'll switch colors to make it kind of distinct. Skeletal muscle cells run parallel to the length of this entire muscle organ. So kind of like so. Convergent, described as having a broad base on one side and it tapers down to a smaller area on the other side. So roughly a triangular shape. There's the wider base, tapering or converging down to a smaller area. And circular, just like the name describes. There's now a circular ring of skeletal muscle around some orifice, around some opening. So your lips, your mouth are a great example with that circular orientation. And let's go right to the textbook figure here. So what I was trying to draw for a parallel muscle shape, a convergent shape again starts with a broader base, tapering down to a smaller area. This cross section will become important in just a moment. And a circular ring. You notice that its function is to close off and constrict some orifice. So now these latter three shapes here, the pennant shapes, uni, of course, meaning one, the muscle fascicles connect to the tendon, running at an angle to one side, bipennate, branching at two separate angles on each side of the tendon, and multipennate now. If you look at this first line of description, multipennate muscle appears at the surface just like convergent. There's a broader base and it tapers down to a smaller area. The distinction now is in multipennate muscle, the tendon is not simply located on each end of the muscle. The tendon actually branches right through the center of the muscle itself. So if we go back to the illustration for convergent muscle, I mentioned a cross section slicing through the center mass of the muscle. There's no present tendon. And we'll skip forward to multipennate. A broad base tapers down to a smaller area, but through cross section, you could see that internal tendon that would branch through the muscle itself. The description of bipennate, you notice here, if I complete this figure, there would be a tendon up here, and then the muscle fascicles are running off to make this bipennate shape. And uni, here's the tendon, and just running at an angle just to one side. So these 6 common shapes for skeletal muscle. And we'll finish here now with the description of the terms origins, insertion, and actions. The first two truly pertain to the tendons. So what's known as an origin and insertion? These terms are now typically some area on the skeleton. Muscles typically connect to the skeleton. And we see that's the point where the tendon precisely would connect. So again, connecting to the various rami and tubercles and tuberosities of the bones, or in some cases, connecting to the skin for facial expressions, connecting to another muscle. And that's what's in common for the terms origin and insertion, just the tendon points where they connect. The second bullet point here, however, this only applies to our appendages. So the muscles that are on our arms or legs. And the reason being this description, the origin tendon, this is always going to be the proximal tendon. And the insertion tendon would be distal. So if we think about our leg, for example, think about your quadriceps muscle. The proximal tendon of the quadriceps muscle, This actually connects to your pelvis, to your coxal bone. That would be the origin, the proximal side. The insertion is distal, and the insertion for your quadriceps is actually all the way down to your patella and then on to your tibia. So the origin for muscles on the arms and legs, that's the proximal, or you could say superior side. And the insertion is the inferior or distal side. This does not apply, though, to the head, neck, and torso. There's no set origin or insertion point for the head, neck, and torso muscles. So again, this second bullet point only applies to the appendages. However, this third bullet point applies to every muscle in our body. So the action is describing the motion, the movement that that muscle now allows. So in all cases, when a muscle is contracting, muscles first can only pull. That's the force they exert, is a pulling force. And they could only pull the insertion tendon closer to the origin tendon. The insertion has the ability to move. The origin tendon is held stationary. So depending on the precise location of the muscle and thus its origin and insertion, each muscle will have its particular action, and we describe those terms as flexion, extension, elevation, depression, whatnot. And again, the structure, the anatomy, the location of the muscle, that's of course what dictates its physiology, its motion. And here's just a nice little sculpture, another example to think of. Here we're seeing the biceps brachii muscle, the origin of the biceps brachii. This is now on the scapula. It's actually these 2 points, hence the name bicep, these 2 branches, these 2 areas here on the scapula. These are the 2 origins. You notice this is the proximal side or the superior side. And now the insertion, you see this right here on the radial tuberosity. This is the insertion. So this is the distal or the inferior point. The action is always to pull the insertion towards the origin. And in this case, the major function of the biceps brachii is to bend the elbow, or the term is flex the elbow. And here's just another figure showing that. So pulling the insertion tendon, applying that force to pull the insertion tendon and physically pull it closer to the origin point.